Okay. So I think both of you done a great job in answering the paper 4. Yeah. I see you know the concept very well. Yeah. So I just have some little corrections about your answer only. Okay, you can see, especially for answering the biology, you need to be precise. For example, okay, you can see this answer. Suggest why a protective substance is necessary in the intestine. So you say that prevent self-digestion. Yes, it's correct, but you can mention prevent self-digestion by what kind of substance. So it is by the hydrochloric acid, right? Because as we know that hydrochloric acid is very, very acidic, and that it will able to corrupt about the stomach lining. Yeah, so you can add cell digestion by hydrochloric acid, and then this protect the intestine infections from the pathogens. Yeah, so pathogen you can mention which is from the virus or bacteria. So this is another answer that you can write for these questions. For example, like prevent the physical damage by the foods when they are passing through the intestine. Yeah. Okay, so this answer journey is okay. It's okay for me. So if I didn't put any comments, means that the answer is correct. Okay. Yeah. I forget the mind. Okay. It's okay. Means the answer is correct. If I don't put any comments. Yeah, this also both correct. Okay, for this. Yes. I think both of you get the correct answer for these two. Okay, but I can add on something. As we know that because at 0.8 molar compared to 0.6 molar, 0.8 molar, you know that is much more we call as hypertonic. Okay, hypertonic means is they have a higher solute potential, higher solute concentration. Yeah, so you can mention that there's a greater water potential gradients in the 0.8 compared to 0.6 mmdm per cube. So this is example of question you can write, but generally I see your answer, I think it's okay. You can score a full mark. And then both also fine, okay. Turgid cell and vacuole is swollen. So you can add on like cell wall. I mean the cell wall will bulging up. And then the cell membrane press on the cell wall. So they will create the turgidity. They will create the tubular pressure. And then for the last one, okay, how active transport different from the osmosis process. So whenever the question asks something like this, so that means that you should write the point about the active transport only, but not the osmosis. You see, the point is just about active transport. So your way to answer is correct. Okay, you say that active transport, they require the use of energy. Okay, energy you can mention. Energy from where? So energy is being produced from the cellular respirations. Okay, in where? In the mitochondria. So see, your answer be specific if you write in this way. So you say that transport is again the constant gradients involve the protein carrier. Yeah, that's correct. So maybe you can say that transport of what kind of the substance. So that means that it involves the transport of the ions or the sugars against the constant gradients with the help of the carrier protein. So this is the example answer you're able to write. Yeah. Okay, so that's all for the chapter 3. So we'll continue to discuss about the new chapter, which is chapter 4, because biological molecule. So you may open this note, biological molecule. Okay, if you're ready, let me know. Yeah. So what is meant by biological molecule? Biological molecule is known as the molecule which can be found from our body. So we know as biological, as we are a biological made organism, right? So you are generally learning about 3 kind of molecule, which is from the proteins, okay? Proteins, fats, and carbohydrates. And then you will realize that the food that you eat every day, include your rice, meats, fish, egg, are all comprised of this carbohydrates, fats, and protein. So we are generally made of from our food that we eat every day. So what is meant by the biological molecule is most of the molecule, they living organisms fall into 3 categories, which is this, okay? And all of the biological molecule, they contain the carbons. So they are also described as the organic molecule, because that we learn about the organic molecule and also the inorganic molecule. In Chinese, they name it as 有机. Okay, inorganic molecule means 无机. So why that means as organic molecule is because they contain these carbons. Carbon is very important for us because in the living earth that we live on, usually the organisms, they are made from carbons, okay? So where is carbon? Carbon is about this thing, C. See, when you have a look on this glucose molecule, so you realize that their molecule is made from the carbons. Here is it. C, 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 C. So if you want to write the chemical formula for the glucose, yeah, so it should be like in this way. C6H12O6. Okay, yeah. Oh. So as you can notice that all carbohydrates, proteins, and the lipids, they are all made from the carbons. See, they all contain the carbons. And also, they do contain the carbon, the hydrogen also, and also the oxygen. So they have the carbon, hydrogen, and oxygen for both one, for these 3 biological molecules. But the difference is for the proteins, okay, they contains other kind of element, which is the nitrogen, sulfur, which is they are not found in the carbohydrate and lipid. Please always remember that carbohydrates and lipids, their structure is almost similar, but the protein is very much more different. So now we talk about the carbohydrates. Carbohydrate is the most, common biological molecule found in our body. So this is the example of the carbohydrates, which is the glucose. Okay. Glucose is a kind of simple sugar, because when we study about the carbohydrates, we should study about 3 class of the carbohydrates, okay? 3 class, which include about the, you can follow me right now, this one. Monosaccharide. Okay, and then the disaccharide. And then the polysaccharide. Those are the 3 molecules for the carbohydrates. So what is meant by the monosaccharide? Monosaccharide is known as the monomer form of the molecule. Monomer form means that they only contains 1 unit only. For example, 1 unit of the glucose, 1 unit of the glucose, 1 unit of the fructose, 1 unit of the galactose. Just 1 unit. Then disaccharide, they are comprised of 2 units of the monosaccharide linked together. So they are made of 2 units. So they are called disaccharide. So disaccharide example, it will be the maltose, sucrose, and lactose. Maltose are the, we call it as a found in the food, we call it a 麦芽糖, okay, maltose. And the sucrose, they are found in the table sugar. Yeah, they are consumed by us. And the lactose are found in the milk. In the milk, they have a lot of lactose. Lactose means 乳糖. Okay, so the polysaccharide, then how about polysaccharide? Polysaccharide is much more complex than the monosaccharide and disaccharide, because in the polysaccharide, they are made of the monomer, which is about 100 above, 200, 300. So there are many units connected together to form a large complex molecule. So this is known as polysaccharide. Remember when you learn about this one, mono means 1, di means 2, Poly means many. This is the English term. Can you give me an example of the carbohydrates which you're able to eat every day in the food? Like the rice, right? Bread, or the noodle. So those are the foods which contain the rich source of the carbohydrates. Yeah. So you get the carbohydrates from those kind of food, which is they contain a rich amount of carbohydrates. So in your rice, noodles, bread, why you are eating them? Because you want to get the chemical energy stored inside the food. Yeah, as the food such as the rice, okay? They contain what? Rice contain the starch, right? Starch. Okay, starch are found in the rice. So when you eat the rice, okay, actually inside contain the starch. So the starch will start undergo the digestion process. So digestion process, which is they will add it with the water, starch plus the water, and then they will break down, okay, by the enzyme. The enzyme help in break down the starch is referred to the amylase. So in our mouth, we have the saliva. So the saliva contains the salivary amylase. So this salivary amylase will help to break down the starch, become the maltose. See, become the maltose. So you can see that from the polysaccharide, which is the starch, they are being break down by the enzyme. So they become the disaccharide, which is maltose. So forget to mention to you that the example of polysaccharide is polysaccharide, is the starch, glycogen, cellulose. So those are the three examples of polysaccharide that you'll be learning. Yeah. So this is the story about the carbohydrates. You just imagine that. Okay, you just imagine that the carbohydrates, okay, the monosaccharide is a building block, right? It's a building block for a house. Because if you want to build a house, which is very big house, you need thousands of the building block. So this monosaccharide is a building block for you to building up a large polysaccharide. Yeah, so maybe we can have a look on the structure. Yeah, so as you can see that this is the structure for the glucose, which is a monosaccharide. So when you are joining two monosaccharide, which is a two glucose together, when you're joining two glucose together, you will form a maltose, which is a disaccharide. So when you join more and more glucose together, you will form another polysaccharide, which is you can form the starch, glycogen, and cellulose. So when the disaccharide, they will appear in this way. You can see that this is the maltose. So maltose is being formed due to the glucose joined to another glucose by the condensation. So condensation is the process which is they will join two different unit of the monosaccharide together to form a one disaccharide unit. So this is condensation. Condensation means combine. It means combine. So like this unit we know as the disaccharide. Yeah, disaccharide. Yeah. So this means one disaccharide from two monosaccharide. So when you join more and more, you will form a polysaccharide. So example, you form it like this. This is the polysaccharide structure. You can see the glycogen, the starch, and also the cellulose. Okay, so those are the we call as polysaccharide. Because they are made from many units of the glucose joining together. So you should know about the usage for those we call as the glycogen, cellulose, and the starch. You should know about these three usage, glycogens, cellulose, and the starch. Okay, for example, glycogens, you see, glycogens are always found in two sites in our body, which is the liver and also the muscle cell. glycogen. So why? Because glycogen is a kind of the energy storage compound. It's an energy storage compound for human. Okay. So I'm not sure, do you still remember about the story that I mentioned to you before, which is the cellular aerobic respiration? So the process is like this, okay? So after you eat the rice, for example, after you eat the rice, so the starch, okay, the starch already been digested to the maltose. Then from the maltose, they will be digested, become the glucose. Okay, so the glucose is a most simplest monomer. Simplest monomer that our body will absorb the glucose to generate the energy. So will be absorbed by the intestine, okay? Because the food absorption are actually done in the intestine. So this will discuss in the chapter 7. Absorbed by the intestine villi. So the glucose will enter bloodstream. So this means that whenever you just finish your dinner, maybe after 1 hour or within 1 hour, your blood glucose level will be rising up. It will be picked up, okay, because they are just finished their digestion. So entering into the nutrient absorption process. So most of glucose will be absorbed, okay, and enter into bloodstream. So from the bloodstream, they will diffuse, okay, they will reach the body cell. Means the body cell will uptake the glucose. So you can imagine it this way. This is the body cell. This is the body cell, right? With the nucleus. This is nucleus. And then this is a cytoplasm. So when we talk about the body cell, majorly we talk about the eukaryotic cell. Okay. Eukaryotic cell. Eukaryotic cell. Okay, Ethan Suyang, may I ask you a question? Eukaryotic cell, they do condense mitochondria or not? They have or they don't have? They have mitochondria or not? Do you still remember mitochondria? Hi. Do you still remember mitochondria? Can you listen to me? Can you hear me?
Yes.
Okay. Eukaryotic cell, they do contain mitochondria or not? They contain or they don't have? Come on.
Yes.
Yes, they have. Okay, thanks. Okay, because previously we discussed about eukaryotic cell and prokaryotic cell, right? Eukaryotic cell is referred to the, I'm not sure, do you remember? They are referred to the animal, plant cell, and the fungus. All of them, they have the mitochondria, except prokaryotic cell. Prokaryotic cell is belong to bacteria. Bacteria, they don't have the mitochondria. Okay, bacteria, they don't have the mitochondria. So usually bacteria, they will be doing we call as the anaerobic respirations. However, the eukaryotic cell, because they have the mitochondria, so they are able to conduct, we call as the aerobic respiration. Aerobic respiration. So they able to conduct this process, which is they're able to use this C6H12O6, which is glucose, together with the oxygen, right? So they will break down the glucose. So they will release the carbon dioxide, water, and also they will release the energy. Here, energy. See? So this is the equation for the cellular aerobic respiration. And remember to draw about the balance, the balance equation, 6. So you can imagine that this is the cell, right? So the glucose from the bloodstream, the glucose, which is this 6 carbon molecule. So they will get into the cell. Yeah, they will get into the cell. So there will be uptake by the mitochondria in the cell. So the mitochondria, they're able to break down the glucose to release the energy. So the purpose of taking up the glucose is they want to break down the glucose. Okay, they want to break down glucose to release energy. Oh, sorry. This is their end. Okay, this is their end. To release energy. So why they need to release energy is because that our cell always require the energy input. So the glucose, will uptake by mitochondria to release energy. Break down to release energy. So the energy will be used by the cell to perform their cellular activity. So this is the purpose, that's why they require the energy. So this is a concept for you. Okay, so the food that you eat every day able to provide the glucose to you. So the body cell able to get the glucose, right, to get the energy. So whenever, if there's an excess glucose being intake into your body, but the glucose was not being used up by the cell, right? So this will cause that the glucose will be staying in your bloodstream. So if the glucose, excess glucose, now we talk about excess glucose, not used up by body cell, okay? So they will be stored in glycogen, okay? They will be convert by insulin. Okay, insulin is a kind of hormones produced by the human pancreas. So the insulin will convert the glucose become the glycogen. See, glycogen. So after that, the glycogen will be stored in our body. Stored in liver and also the muscle cell. These two places. Yeah. So why they are doing this? Because they want to reduce the blood glucose level. Okay, if you have excess glucose are not being used up by your body cell, but the glucose will be stay alone in your bloodstream. So this will cause a constant elevations, increasing of your blood glucose level. So this will cause a very bad impact to your health. Okay. Usually the diabetes person, they are unable to control their glucose level. So they are required the artificial injections for the human insulin. Okay, so this is the glycogen. So glycogen, you know that it's a kind of storage compound in the liver and muscle cell. So whenever when body need glucose again, maybe you already few days didn't eat, so you force the body to burn the glucose source. So when the body require the glucose input, so the glycogen will convert back into glucose and release to bloodstream. So this will cause the blood glucose level to increase. I hope you understand this concept. So because you need to understand the concept first, so you are feeling that you are not memorizing. So this is the reason why the glycogen, they appear in this way. See, very large, complex polysaccharide. When you have a look, it will be something like this. See, this is glycogen. Okay, actually, I got insert this picture in the graphic, in the graphic document for the chapter 4. You can have a look. So this is glycogens. So this small subunit, they are actually the glucose unit. So the glycogen is a polysaccharide. So glycogen is a storage compound. Okay. Then now we talk about the starch. Yeah, this is a starch. Starch are found in the plant. Okay, for the glycogen, they are usually found in animal like us, like the dogs, cats are the animal. Okay, for your information, the plant will store their excess. Plant store excess the glucose in form of starch. Okay, plant they require the nutrients molecule to help them to grow also lah. And then the glucose source from the plant is come from a process which is called as photosynthesis. Okay, because photosynthesis that is a equation. Okay, is a process that they are converting the carbon dioxide. Yeah, they are converting the carbon dioxide plus the water, and they will be producing the glucose and releasing the oxygen. So this photosynthesis, this is a equation of photosynthesis. So the outright will be the sunlight. Sorry, my pen is not functioning. Yeah, why? I think I need to type. Sorry. Yeah. So the outright will be the sunlight. So the below it will be the chlorophyll. So those are the equations for the photosynthesis. So photosynthesis is a process which is they will produce the glucose in the leaf. So photosynthesis, they will produce the glucose in the leaf, right? Leaf. Okay, because that leaf, they do contain the chloroplast. Leaf contain the chloroplast. I mentioned before, right? There are three types of plant cell, they contain the chloroplast. I'm not sure you still remember or not. In the chapter 1, I mentioned before. 3 kind of plant cell contain the chloroplast. Do you want to give me an example? In the leaf, okay, which is the palisade mesophil cell, spongy mesophil cell, and the gut cell, right? So this is a leaf, they do contain the chloroplast, so they're able to perform the photosynthesis to produce the glucose. So leaf is a kind of, we call it source. I will learn more later. So leaf is source. Source. Because they produce glucose. Okay, however, the difference with the human is in the animal, after the leaf produces the glucose will be converted into the sucrose. Okay, so they convert it into sucrose, they will be transport to the phloem. Do you still remember the plant structure? They do contain the phloem. Phloem, they help in the translocation. Okay, just remember that they are helping in the translocation. It's just like how the plant able to transport their nutrient molecule a long way in the plant body. So they are using the translocation to help them to transport the nutrient molecule from the source of productions to the place which is they require the nutrients. So this is the story about the plant. Okay. However, if there is an excess sucrose, they are not being used, so they will be stored as starch. See, they will become starch and store in the sink. Okay, in the roots. Yeah. So starch is a storage compound for plant. So you can clearly see that in the animal, they usually store their excess nutrient in form of glycogen, which is found in the liver and the muscle cell. In the plants, they will store their excess nutrient in form of starch, which is found in their roots, or we call it zinc. which is the place that they store the excess nutrient. Then however, then how about in the cellulose? Cellulose is the component for the cell wall. Cellulose make up the cell wall. Okay, so that's why the cell wall, they are become much more rigid. And they are very compressible, not easily ruptured. They help to prevent plant cell from rupture due to the high tensile strength by the cellulose. So this cellulose will create a high tensile strength for the cell wall. So it will prevent the cell wall from being burst. Yeah. So this is the three polysaccharide, their usage and the functions. Okay. So all of them are being made from the glucose molecule, which is this glucose molecule. Okay, this is for you to have a look, yeah. How to separate glucose molecule able to combine together to form a one unit of disaccharide. So this process we call as a condensation. Combine, okay? Condensation means combine. And then with the production of the water molecule. So however, if you want to spill, okay, if you want to spill this disaccharide back to become the monosaccharide, like this. Okay. If you want to spill this disaccharide, what happened to my pants? So yeah, my drawing table have some problem. Okay. If you want to convert, for example, you have a look. If you want to convert this, we call as the disaccharide, yeah. You want to convert this disaccharide, which is a maltose, back to become the monosaccharide. From the disaccharide back to become monosaccharide. So it can be done. Okay, just that the process we call as the hydrolysis. Hydrolysis, which is involving in the conversion of the disaccharide become the two separate monosaccharide. So the condensation is they will be involving the combination for the monosaccharide become the disaccharide. So the upside will be the combination. Oh, sorry, condensation. Yeah. So I hope you know about what is meant by the condensation and the hydrolysis. Hydrolysis means build up, separate. One unit become two separate units. So we call it hydrolysis. Lysis means separate. Hydro means that they use the water to separate. Okay, as you can see that disaccharide plus the water, so they will become two monosaccharide. That means that they are using the water to help to spill the molecule. Okay, so this is example of photo for you to have a look. Galactose and glucose. In your stage, you don't need to know too detailed about the structure. You just need to know about what kind of the molecule are found in the monosaccharide, disaccharide, and the polysaccharide. You don't need to know too much detail about structure. The structure will be you'll be learning when you are going to higher level study like the A level. Yeah. So this is for you to have a look. Sucrose. So this is sucrose, yeah. So sucrose are the combinations, okay, which is a product of the condensation from the one glucose molecule and another fructose molecule. Glucose plus fructose, they will form one sucrose. And then this one, we will talk about the lactose. This is an example of lactose. With the condensations between one galactose unit to one glucose unit, so they will form one lactose, which is disaccharide. Yeah, so they look in this way. Two molecules linked together by the bond for the disaccharide. So I think that's all about the carbohydrate. Okay, we move to next one, which is the fats. Yeah. The unit of the fats we call as a triglyceride. So triglyceride is made from one glycerol and the three fatty acid. One glycerol and the three fatty acid, which is this one. This is one glycerol. Okay, this is three fatty acid. 1 glycerol and 3 fatty acid. And then the fat actually are being divided into two kind of the physical form. Okay, when we talk about the solid, example of solid, we call it as a butter. Okay, the butter that you spread on your bread is a solid form of fat. Another liquid form of fat would be something like cooking oil. Okay, so that means that one appears as solid at the room temperature, another appears as liquid at the at this room temperature. So this is all about the fat. Okay, so the fat you should know about the structure. Okay, you should able to identify the structure. So this is glycerol. So 1 glycerol and 3 fatty acid. The glycerol is just made out from the hydrocarbon. Okay, the chain which is from the three carbons. For the fatty acid, it will be much more complex. So the 3 fatty acid, as you can see that they are very long hydrocarbon chain. They will describe a very long hydrocarbon chain. So this long hydrocarbon chain, actually, we call it as non-polar. Okay, Ethan Suyang, let me ask you, do fat or oil, okay, able to dissolve in water? Do the triglyceride able to dissolve in water? Can they dissolve in water or not? Yes or no? Yes or no? You can imagine that can oil dissolve in water?
No.
No, yes. No, definitely it's not. Do you know why they cannot dissolve in water? This is because that they have this very long hydrocarbon chain. See, so long. How many carbon? 1, 2, 3, 4, 5, 6, 7, 8, 9. Oh, and then you have 2 more. So it's so much long hydrocarbon chain. So this hydrocarbon chain is non-polar, definitely non-polar. So non-polar means that they are unable to form hydrogen bond with water. So that's the reason that the triglyceride just unable to dissolve in the water. Okay, so it's insoluble. Yeah, it's insoluble. So for example, fat, you should able to recognize the structure. See, 1 glycerol plus 3 fatty acid. Fatty acid is they have 3, okay, R with the COOH. Because fatty acid, they do contain the carboxylic group. So this COOH we call as a carboxylic group. Actually, fatty acid, for information, that fatty acid is actually acidic. It's an acidic pH, because they do contain this COOH group, which is carboxylic. So 1 glycerol with 3 fatty acids, so they will form the triglyceride. So this process we call as the condensation, which is combinations of 1 glycerol and 3 fatty acid, or we call as the esterification. And then the glycerol and fatty acid, they are joined together by the bond we call as an ester bond. So the ester bond is found between where? Between this C and O. See, this one. This is the ester bond here. E. Ester bond. Okay, so that's all about lipid. And then now we continue to learn about the proteins. Okay, another interesting topic about the protein is proteins. They help to make up of our body cell, the organelle, right? And also our muscle tissue. So protein is actually made from the amino acid. This is amino acid. We will talk about amino acids, okay? Actually, we learn about this structure. Okay. 1 amino acid join with another amino acid. So 1 amino acid, they do contain we call as the amine group. This is the amine group. NH2, we call it amine group. And then COH group, we call as the carboxylic group. COH, you can see, carboxylic group. So one amino acid, they are classified as alkaline or acidic because they have one amine group and one carboxylic group. So when you join to another amino acid, okay, you join by where? You are joining by the amino acids from the carboxylic group to another amine group from another amino acid. So this process will help to produce one water molecule. See, they will produce one water molecule. Oh, sorry. So actually, so at the end, they will form a dipeptide. So with the production of the one water molecule, yeah. So this we know as a condensation. You can see one water molecule is being thrown out when you are trying to join these two amino acids together to form one dipeptide. So whenever you want to break down the dipeptide back to their two separate amino acids, you just add in with the water. So we call it a hydrolysis. You just use the water, add in, help to spill the dipeptide become two separate amino acids. So the bond they help to link between the two amino acids, they call as a peptide bond. See, this is a peptide bond, which is between the CONH, yeah, which is they draw in the yellow color. We call it peptide bond. Okay, peptide bond are being break down by, okay, peptide bond is very strong bond. Because peptide bond is a covalent bond. Covalent bond is very strong. Strong bond. Whenever I mean strong bond, means that this bond is very strong and is does not very easily affected by the change in the pH or temperature. By the temperature or pH. But there's one kind of substance able to break the bond. So they're able to lyse the bond for this peptide. Okay, they're able to break. They're able to break it. So the substance we call as the enzyme. Only the enzyme able to break the peptide bond. Only enzyme, which is the protease. Okay, there's a group of enzyme digest a protein we call as protease. Only enzyme protease able to break down the peptide bond. Okay, so this shows that peptide bond is really very strong. So it's not just like other bonds like the hydrogen bonds. They are very easily affected by the change in the temperature or pH. Means that is the temperature too high or too low, or the pH too acidic or too alkaline, they will affect, they will help to break down the bond. So remember, peptide bond join two amino acids together to form one dipeptide unit. So by the condensation. So in our body, we actually have 20 type of amino acid in our body. And then with the different R group, okay, different R group. So the amino acid, because the amino acids, they will be joined together, then they will form the proteins. Okay, so I hope you still remember about these rules. Okay, you can see. This is the amino acid structure. So how protein is being made of? Protein is being made from the amino acids. So amino acid will be joined together. They will form the peptide. Initially, they will form the polypeptide change. Polypeptide change. This one. Polypeptide change. So amino acid, they will be joined by the peptide bond to form the polypeptide change. So you can see that this peptide bond, they join from one amino acid to another amino acid, and then to another amino acid. So they will form a one change, we call it as polypeptide change. Okay, so after that, when this polypeptide change, they will be undergo folding. Okay, they will further folding, or they will be joined with another chain, another polypeptide change. So maybe 2 or 3 polypeptide chain, they will join together and then they will fold. Okay, they will fold into, we call it as the globular shape. or we call as three-dimensional protein structure. What is meant by this global shape? Global shape means that shape look very round, something like a ball, 球体. Okay, that means 球体. Because when we talk about protein product usually their structure is ball shape. Ball shape. Or we call it as three-dimensional structure. So they will form the protein products. Okay. Okay, this you can have a read. You can see that because in the proteins product that we learn in this IGCSE syllabus, there will be few. Okay, for example, the protein product they will be learning protein product. Okay, produced by the cell. So the responsible for the body cell is they will help to produce the protein product. So example will be the enzyme. Enzyme is one type of protein product. Okay, you can say in this way. All enzymes in your body are made from proteins. Yeah. And then another more, hormones. Another more, antibody. So those are three substances which is they are belong to the proteins products. And then this tell you that many of the proteins they have a different shape, and the shape has affect the protein's functions. And then for example, the enzyme, okay, enzyme you will learn in next chapter. So they have the active site. So this active site, they have a specific shape. So to allow the substrate with the we call it complementary shape. So whenever the substrate able to fit into the enzyme active site, so they're able to form the we call as enzyme substrate complex. Yeah. So this means that the enzyme active site must be we call as able to fit into the structure of substrate. Or we use another term, okay? The enzyme active site must be we call as complementary, okay? Complementary to the structure of proteins. So it's just something this way, okay? Enzyme substrate like this. This is the enzyme. You know what is enzyme? Enzyme we call as 酵素 in 华语. Oh, sorry, I cannot draw it. Maybe I show some photo. Better for you to show some photo. I show some enzyme substrate photo. Okay. Do you able to follow me currently? Adam Suyang? I'm not sure am I speaking too fast or not. Everything is fine?
Yeah.
Okay.
Yeah.
Okay, sure. But I hope I can get some question for you, okay? You can ask me any question. I'm sharing the screen, right? Yes, I'm sharing the screen, okay. Yeah, this is example of the enzyme substrate. Yeah, enzyme. In your life, you can see a lot of enzyme. For example, the biological detergents that your mother are using, okay, they also contains a lot of enzyme. So this is an enzyme. Okay, enzyme, the function is they will help to increase the rate of chemical reactions. Oh, sorry, enzyme. Enzyme, they will help to increase, rate of chemical reactions. So that means that if there's a chemical reaction happen without the enzyme, so the chemical reaction will be happen very, very, very slow. So the chemical reaction is they need to convert, okay, they need to convert the substrate become the products. So enzyme help to convert the substrate become the products in a faster rate. So that's why we need enzyme. Especially in the digestive process, Just how we mentioned about the digestion process, right? Which is after we eat the rice, so the rice will be catalyzed by the enzyme in our saliva. We call it the salivary amylase. So those are the enzymes. So enzyme, they are a protein's product. So enzyme, they have a specific shape on their active site. This, okay, this is active site. So they have a specific shape on the active site. So you can see that whenever the substrate bind to the active site enzyme, this means that the substrate must be able to fit, okay, fits into active site enzyme. So they're able to form the enzyme substrate complex. So the enzyme able to perform their chemical reactions. So they're able to change the substrate, become the products. So this is we call as an enzyme catalyzed reactions. Enzyme catalyzed reaction. Okay. So every enzyme, they have their different shape for the active site. For example, you have three kind, or maybe you have example of two enzymes, right? Protease and lipase. Okay. Protease is help to digest the proteins. Lipase is they're helping to digest a lipid. So the structure for the protease and this lipase, even though they are also enzyme, okay, they are enzyme, but they have the different shape in their active site. So that's why protease only able to bind to the proteins, and the lipase only able to bind to the lipid. And then another one, antibody, okay? Antibody in Hawaii we call it 抗体, okay? So every time you listen to your family, always tell you that you must be strong, okay, and healthy. So a strong and healthy person that have a high level of antibody in their body help them to produce, help them to protect them away from the infection of the disease. So antibody. So antibody is a kind of protein also. Antibody is actually produced by the white blood cell because it's lymphocyte. Lymphocyte. Yeah. So the antibody, whenever they need to bind to the pathogens with their antigens, so it's something like this. I find you some picture. We call it as antibody antigen complex. Antibody antigen complex. This one. Yes, you can have a look. This Y shape. I don't know why I cannot draw on my laptop. I'm so sorry about this. This Y shape, okay, is antibody. And this pathogen, this yellow color is a pathogen, okay? And then this blue color found on the pathogen is referred to the antigen. So you can see that this antibody able to bind to the antigen. So they will form the antibody able to bind to antigen on the surface of pathogen. So they will produce a antigen antibody complex. So this is happened during the we call it as an immune defense mechanism when your body are helping. They are trying to fighting the pathogens, helping your body to clear away the pathogens. So yeah, so this is the shape of antibody. Yeah, see, the shape of antibody must be always matching to the shape of antigen, so they can attach to it, and then they can help to destroy the pathogens. Okay, so whenever we talk about the protein shape or structure, please remember always about this rule. Okay, maybe I can show you a picture again. How the protein is being produced from body cell is they are originated from the DNA. Okay, I hope you still remember about this picture. Yeah, this picture about how the proteins produced from the DNA. This picture. It's very important picture. This one. See? Oh, sorry. Okay, we learned this before, right? This is inside the cell nucleus. Inside the cell nucleus, they have the DNA. DNA. Okay, what is DNA? DNA, they store the genetic information. Yeah. Then inside the DNA, they do have a lot of gene, right? So they contain the gene. What is gene? Gene means 基因, okay, gene. So gene is a length of DNA help to encode for proteins. So you can see that this is the DNA, right? DNA strands are open up. So when they open up, they will expose themselves to the, we call it as a outside, okay? So they will produce a mRNA, we call it as a messenger RNA. So the messenger RNA, after they are being produced by the transcriptions, so the messenger RNA, they will help to copy, their role is they will copy the genetic information from DNA. So after they copy the genetic information from the DNA, then there will be, okay, so they already contain the, we call it the nucleotide base sequence, right, for the mRNA. So they will bring this information out from the nucleus into the cytoplasm. So in the cytoplasm, mRNA, they will bind to the ribosome. Why? Because the ribosomes is, they will help to, okay, do you know what is the function of ribosome? Function of ribosome is they will help to do what? Whenever the exam asking you what is the function of ribosome, what answer you should write? They will help to
Read the genetic information and create the amino acids.
Yeah. Correct. Okay. Sorry, your sound is very low a bit, but I heard amino acid. I think you get the correct answer. Okay, but you just write synthesis protein. Yeah. Okay, but you say that amino acid is correct also. Maybe you can say that they are trying to link the amino acid together to form proteins. So this is the ribosome. So ribosome, they will be bind to the mRNA. So the mRNA, they will be like in the middle of the ribosome. Because ribosome, you are contain two subunit, right? One is a large subunit on top of it. This is because large subunit. And then below there will be small subunit. Ribosome, they are comprised of two subunits for your information. So ribosome, their role is they will help to read along the information on the messenger RNA. And then what they do, they'll be calling tRNA to come. What is mean by tRNA? tRNA means the transfer RNA. tRNA means the transfer RNA. Transfer RNA. So you can see that transfer RNA, they will be carry the amino acids. So you have 20 amino acid, right? You have 20 type of amino acid in your body. So that means that you have around 20 times 3. You have around 60 of this transfer RNA. So because transfer RNA, they always carry with the amino acid on top of their head. So they were calling the transfer RNA to come. And then this transfer RNA, actually, they are contains 3 base sequence that you can see. These are UAC, CAC, GCA. So they will be pairing of the base between the messenger RNA and also the transfer RNA. So the transfer RNA able to carry with their respective the MSC coming. So by this way, they able to form the polypeptide change. By joining the peptide bond, see, they are joining the peptide bond between the amino acid in the ribosome. So this is the way they are able to produce the proteins from the DNA. Always in this method, transcriptions and translations in the ribosome. So okay, you just remember about this formula. Okay, I write it down. You can see that how body cell produce protein. This is a kind of question coming out in the IGCSE exam before. So you can write it in the base, this one, nucleotide, see, nucleotide base sequence of the DNA, right? So for the nucleotide base sequence of DNA, they will be becoming what? Become the messenger RNA. Or you can write mRNA. Then they will enter from where? From the nucleus. Then they will get to the cytoplasm. They will get to cytoplasm. And then the cytoplasm, they will be approaching to the ribosome. Ribosome will be carried out what? They will be carrying out the translation process. So please remember to write these translations in your answer. So the ribosome, okay, they will be producing the polypeptide change. So this has shown that the amino acid sequence is being determined by the nucleotide base sequence. Okay, amino acid sequence is being determined by nucleotide base sequence. Yeah, remember that. So from the different amino acid sequence, different amino acid sequence, they will be creating a different protein structure because protein structure are being created based on the sequence. Structure or shape. Structure or shape is similar meaning. Yeah. So then the protein structure or their shape will determine the protein functions. Yeah, so this is story about the proteins. Okay, so I hope you understand this. See, because that different sequence of the amino acids, they will cause the chain to fold in different way. You have 20 type of amino acid, right? So when they arrange in different sequence, they will cause different structure will be produced. So different structure produced, it will carry out its own functions. So every kind of proteins, they have a unique 3D shape, means that three-dimensional shape. Three-dimensional shape. Okay, so for information, okay, for example, if you compare with the lipase with another lipase, they have the same structure. But if you compare with the lipase with protease or amylase, they have same structure or they have different structure? Same or different? Lipase with protease and amylase, do you think they have same structure? Ethan Suya, do you think they have same structure? I'm waiting for your answer. Hello? Do you think the lipase, protease, and amylase, they have the same shape or not? Hello?
Yes.
No, the answer is no, yeah. Lipase, protease, and amylase, they don't have the same shape. Do you know why? No, no. They are not same structure. Do you know why? Because protease only able to bind to protein, right? And then lipase only able to bind to lipid. Protein is a substrate. This one, protein is substrate. Protease is the enzyme. Protease is the enzyme. Lipase also. Lipase is the enzyme. Lipid is the substrate itself. And then amylase only able to bind to carbohydrates. Yeah. So protease, lipase, and amylase, definitely their structure is very, very different among three of them. Different. So because the structure is different, they cause that only protease able to bind the proteins. So that means that protease unable to bind to lipid or carbohydrates. Okay, understand? Protease unable to bind to lipid or carbohydrate because the structure is not same. Lipase able to bind to lipid, but lipase unable to bind to proteins or carbohydrates. Amylase able to bind to carbohydrates, but amylase unable to bind to proteins or lipid. Can you get me? Adam Suya, can you get me?
Yeah.
Okay, so yeah. So this means that every enzyme have their own unique structure. The conclusion, see, every enzyme or every protein, they have their own unique structure. So they are able to carry out these functions. So so it means that the enzyme with the specific active site able to bind to the substrate, which able to fix in the active site. Antibody with their specific sites, okay, only able to bind to the antigens on the pathogens. You can imagine that, for example, if you get the two kind of disease, okay, if you are infected with these two kind of disease, for example, you are infected with COVID, and the influenza A, both also virus, right? You are infected with COVID virus and the influenza virus, for example. Very unlucky, you are very unlucky. You are being infected by these two kind of different virus in your body. So this COVID virus, they will induce your body lymphocyte to produce the antibody just only for the COVID virus. Okay, so this influenza virus, they only induce your antibody, they induce your lymphocyte to produce an antibody for that specific influenza virus only. So that means that the antibody for the influenza virus unable to bind to COVID virus because their shape is not fixed. It's not complementary, okay? It's not fixed. Then that means that the COVID virus, the antibody, see, this antibody able to bind to the COVID virus, but they are unable to bind to influenza virus because they are not fixed. So in the proteins, okay, we always talk about the specificity. So this is called as the specificity. Specificity. This one, okay. This one, specificity. Enzyme must be specific in their action. Okay, they must be specific in their action due to their shape, due to their structure. Okay, such a long story about the protein. Yeah. Okay, for your information, the proteins, are being classified as 4 level of organization of proteins. 4 level of the organization of the protein. So you can see from the primary structure, which is the polypeptide chain. Primary structure is a polypeptide chain, right? Polypeptide chain. So, and then this polypeptide chain, they will be folding, become the secondary structure. There are two kinds of secondary structure for information. We call as beta pleat and alpha helix. Okay, maybe I show you some photo. Wait me for a moment. Alpha helix and beta pleat. I hope you understand that this learning about this protein or enzyme biology is very, very important and it's also very, very interesting because it's very realistic. Yeah. This one. Because sorry, I didn't have. Okay, I will put this. This one. This is the example of the second structural enzyme. See, alpha helic and the beta pleat. See about the structure. This is actually the polypeptide chain, right? So when the polypeptide chains, they are able to connect with another chain, so they will form the bond. They will form the bond. Okay, so after second structure, then we talk about the tertiary structure. These tertiary structures, they will start to form about the structure of the enzyme. Form the enzyme structure by the tertiary structure. So what is meant by tertiary structure? Tertiary structure means that maybe there is one or more polypeptide chains, they are being joined together and they are being linked together by the bond. Okay, for example, like this. Okay, this is a picture. You can see that. From the primary structure, okay, which is the amino acid are being connected by the peptide bond. See, between this lysine, glycine, leucine, valine, like this, all are the peptide bond. So here's a peptide bond between the structure, peptide bond. So until they are being linked together again by another bond, we call it a hydrogen bond. Do learn chemistry, right? In the chemistry, you learn about those bonds. So hydrogen bond, they will help to produce in this kind of shape. We call it alpha helic or beta pleat shape. And then they'll be further coiled together and folded. So they will form this tertiary structure. So see, in this tertiary structure, they will form a globular shape, 球体, globular shape. Yeah, just like ball. Okay, so the polypeptide chain is able to fold due to there's an interaction between the amino acids, due to there's a lot of bond holding between the chains. So they will fold inward together. And then if you have maybe two or more polypeptide chains, if you have two or more polypeptide chains joined to each other, so you form a quaternary structure. Quaternary structure is the most complex form for the proteins. So you can see in this quaternary structure, you have how many polypeptide chains? You have 1, 2, 3, 4. You have 4 polypeptide chains in this structure. See, 1, 2, 3, 4. There are total 4 polypeptide chains in this quaternary structure. So they will form one protein's product. So example of quaternary structures you can study is about the hemoglobin. Okay, let me show the structure for the hemoglobin, our red blood cell, component of our red blood cell. Do I show in this picture? Component of red blood cell. Let me check. I think I didn't show it. Okay, it's here. Sorry, it's here. Okay, I do include this picture in the chapter 4 graphic document. I do include this picture. So you can see this is a red blood cell. So this red blood cell, they are made from hemoglobin. Hemoglobin, that means 血红素 in Mandarin. Yeah, this hemoglobin, actually, they are made from 4 polypeptide chains. Made from 4 polypeptide chains. Which is they have 2 alpha helic and 2 beta pleat. 2 alpha and 2 beta. You can see this is structure. So these 4 polypeptide chain, they will join together. So they will form one hemoglobin molecule. They will form one hemoglobin molecule. And then this yellow color thing is another of the, we call as non-protein structure. Because hemoglobin not only include the protein, part, they also contain the non-protein part. Non-protein. Okay, so the non-protein are included in here is the iron and the oxygens. Those are the non-proteins. So because that hemoglobins, okay, they help to transport the oxygen, right? So they need the iron inside. Okay, so that's all for the protein. I think I finished teaching about the protein already. So I hope you understand. Remember, protein is all about their structure, their shape. So remember about this one. Very important, this one. Okay, I highlight in yellow, means very important. So because in the IGCSE exam, they will ask you in this question, they will ask you that tell me the polypeptide change is determined by what kind of the structure. So you can say that the amino acid sequence is being determined by the nucleotide base sequence. You should understand about the relationship between the DNA and the protein. Okay, so now we enter about the food test. Okay, still have time. Food test. Because that just now we learned about 3 molecules, right? Carbohydrates, proteins, and the lipid. So those 3 molecules are found in our food that we eat every day. For example, the rice, the vegetable, include the fiber, okay, and also the fish, the meat, they got the proteins. So how we test the food, they do contain the nutrients molecule, such as the glucose or the lipid or the starch. So we have those food tests. So for example, if you want to test for the glucose found in our food or not, okay, we have a test we call it a Benedict test. Benedict test, which is this one. This is the Benedict test. Benedict. Hence, so this blue color solution we call as Benedict solution. So initially, this Benedict solution, they are blue color, light blue color. Why? Because they contain copper 2 plus. So copper 2 plus, naturally, they will give this solution with light blue color. So pretty, right? So after that, when they undergo the Benedict test, if there's a presence of the reducing sugar, because the purpose of the Benedict solutions, the Benedict test, is they want to test for the presence of reducing sugar. Okay, test for presence of reducing sugar. So if there's a reducing sugar inside the test, okay, what is meant by reducing sugar? Reducing sugar means that this sugar, this one, in your syllabus, you learn about those sugar, glucose, fructose, lactose, maltose, galactose. Those are the reducing sugars. And then one non-reducing sugar is referred to the sucrose. Sucrose is the only one non-reducing sugar. So what can be done by this reducing sugar? If there's a presence of reducing sugars in the Benedict solutions, which contain the copper 2 plus ion, so they are able to, we call as reduce, become the copper 1 plus. Okay, copper 1 plus, not 2 plus already, become 1 plus due to the reductions, because they undergo the reductions. Oh, sorry, it's not undergo reduction. I think they will be undergo the oxidation, okay? Oxidation. So if there's a, we call as a reduced sugar, which is though glucose, okay, the glucose, fructose, lactose, maltose, galactose found inside the solutions. So this will cause the solutions able to change their color, okay? Initially from the blue, the green, the orange, then at the end become the brick red precipitate. So this is the color that we want. We want to see them become the brick red precipitate. Means there's a large amount of the reduced sugar are found inside the solution. If there's a green or orange color, means that there's only small amount of reduced sugar found inside the solution. So this means that if we put the sucrose, inside the Benedict solutions, can it change to brick red precipitate? Can you change to brick red precipitate if we put the sucrose inside these Benedict solutions, which is blue color? Can it change into brick red color? The answer is cannot. Okay, because it's a non-bleaching sugar. So sucrose cannot. It will cause the Benedict solution remain blue. But the reducing sugar, they will cause the Benedict solution change from blue color to brick red color. So that's the thing that you should know. That's all. So whenever you want to conduct this Benedict solution test, you should heat up the solution in the water bath for 5 minutes. And then you need to insert the sample content inside the solutions. And then you can observe about the color change. So slowly you can observe there's a changing in color from the blue color to green color, then to yellow, then to the brick red precipitate. Okay, so generally the experimenter will be conducted in this way. So you put the Benedict solution, which is initially blue color, into the solution you need to be tested. So the solutions is include any kind of glucose solutions, and then you put into the water bath, slowly heat up. So you boil the mixture about 5 minutes, and then you can see there's a color change. Okay, so some of the safety precaution that you need to aware is you need to handle your test tube with tongue, because to prevent your hand being heat up. Okay, and then you need to wear the safety goggles and the glove when you are conducting this experiment. So this experiment you will be learning in the paper 6. Okay, I'll give you example of the question to do for the paper 6. Okay, now we talk about the protein test. Protein test involved in the Biuret solution. Okay, last one. Protein test involved in the Biuret solution. If there's a protein content inside the food, for example, meat, fish. So when you put the Biuret solutions, which is initially blue color, right? See, Biuret solution is blue color. Same with the Benedict solution, also blue color. So if there's a protein presence inside the food, they will cause the blue color change, become the purple or violet color. So pretty. See, they become the purple color already. So if there's a purple color, that means there is a presence of proteins. If they do not change to purple color, means that there's no protein inside. Yeah, so this is called the Biuret test. And then about the starch, we call it as the iodine test for starch. Okay, what kind of food contain the starch? You can give an example, which is the rice, potato. Those are very rich in the starch. So whenever you put the food sample which is starch contents, when you are mixing with the iodine, they will change color from brown into dark blue like this. See, this is a potato. Okay, so when you put the iodines into the potato, so it will change into dark blue color. Okay, and then Suyang, tell me, what is the original color for the iodine before it changed into dark blue? What is the original color for the iodine? Is what color? It's yellow, red, green, black, or brown? What color for the iodine? Okay, iodine solution, I think you must see before, right? When you are having an injury, you apply the iodines on your wound. So what is the iodine color? Have a guess? 1, 2, 3. Yes. And then Suyang, what is the color of iodine?
Purple.
Purple? Okay, it's okay. The answer is yellow brown. Yellow brown. Okay, this is the initial color for iodine. See, yellow brown. Yellow brown. You can see this iodine in from this. Okay, I show you iodine solution. You must see before. This is iodine solution. So it's a yellowish brown. You can see brown or yellow, up to you. See, this is the color for the iodine. Yeah, this is the color for iodine. So when you put these iodine solutions on the food, they contain the starch. So they will cause the yellow or brown change to become dark blue. Okay, in the presence of starch. So the potato contain the starch. They will cause iodine change color, become the blue black. So they will change from the orange brown to blue black. This is the color you should write. Orange brown. Yeah, that's it. For the so we usually use the iodine to test for the starch in the food sample. So we should know that starch is a kind of polysaccharide, right? Polysaccharide. Starch, right? It's a polysaccharide. So starch are able to digest by this amylase. Okay, amylase is an enzyme to digest the starch. So they will become the maltose. Maltose is a disaccharide, right? Disaccharide. Oh, sorry. Maltose is a disaccharide. Disaccharide. So you can see when you add the iodine solution to the starch, they will cause the iodine solution change from the orange brown become the dark blue color. However, when you add the iodine solutions inside the solutions which contain the maltose, so that means the iodine they just remain in their orange brown color. Okay, they remain their original color, which is orange brown. They did not change into the dark blue because there's no presence of starch. No more presence of starch because they become maltose already. Yeah. So this iodine test only specific in detecting starch only. Not other sugar like maltose, sucrose. No, they only for starch only. Okay, last one. I think we have some time. Okay. Oh. Okay. We studied about. The last test for the lipid. If there's a food contain the lipid, for example, the cooking oil. Yeah. So you should know that lipid is insoluble in water. Insoluble in water, right? So this is we call as an emulsion test. Why? Because when you put the sample and the 95% ethanol and then added to the distilled water, so you will form we call as a cloudy emulsions. Okay, so you should see initially you mix a food sample into ethanol and then you shaken the food sample and then you added this ethanol into the cold water. So you should know that water is not able to dissolve the lipid, right? So they will show the cloudy emulsion like this. This. Okay, this is called as a cloudy emulsion. This is because that water unable to dissolve lipid. So they will form this emulsion. So this we call as an emulsion test. They have a name for this because emulsion test. So if there's a lipid inside the food sample, after they mix with ethanol, okay, because you know that ethanol is we call as a non-polar solvent. They able to dissolve lipid. Just that water unable to dissolve lipid, so they will form the emulsion test. They will form the cloudy emulsion, like this color. Okay, so if you have lipid inside, you produce an emulsion. If you don't have a lipid inside, they will still remain a color in the distilled water. So I think that's all for today. Okay, I'll give you homework later. So see you in next class. Okay, thank you. Bye.